Welcome to our session on Paramyxoviridae viruses. Under the Paramyxoviridae family, we have two subfamilies, Paramyxoviridae with 5 genera and subfamily Pneumoviridae with 2 genera. Under the Paramyxoviridae family, we have the Respirovirus, which includes human parainfluenza viruses 1 and 3, and under the Rubella virus, we have mumps virus and HPIV 2 and 4. We have Morbillivirus genus, which includes measles virus that you are familiar with. We have Nipah virus, which includes Hendra virus and Nipah virus. Now, these are rare viruses in humans. I'm skipping the Avula virus on purpose. Now, the Hendra virus is quite rare, and it was associated with an outbreak in horses and humans in Australia in 1994. Nipah virus was associated with an outbreak of respiratory and neurological illness in Malaysia. But at the time, it was kind of a mysterious virus because the source of this virus was not known. But there was this a scientist who noticed that people who were getting sick with the Nipah virus were those who consume pigs. In fact, he noticed that Muslims were not getting sick with this disease, and he thought, well, maybe those because Muslims don't consume pig, so maybe pig is the source. And in fact, he took a sample, flew to the United States, and identified the virus as Nipah virus. Again, these are rare diseases. Subfamily Pneumoviridae has the genus Pneumovirus, which includes respiratory syncytial virus (RSV) and metapneumovirus (HMPV), human metapneumovirus. What about the properties of these viruses? So they are relatively larger in size, with the virions ranging between 150 to 300 nanometer, being spherical or pleomorphic, and they have a non-segmented or a linear negative sense single-stranded RNA genome. They have envelope, and they replicate in the cytoplasm. Most of these viruses pretty much resemble each other under the electron microscope. Transmission is via the respiratory route by inhalation of respiratory droplets. Now, this is the typical structure of a paramyxovirus. Okay, so as we mentioned, these are envelope viruses, and in the envelope, we have these glycoproteins that are studied and based on which we can differentiate these viruses. So we have the viral attachment protein, which includes hemagglutinin neuraminase for parainfluenza virus and mumps virus. In case of measles, it only has the hemagglutinin protein, the H protein, so they can hemagglutinate blood. For respiratory syncytial virus, we would have the G protein, okay, and we also have on the surface a fusion glycoprotein. For example, respiratory syncytial virus has a fusion protein, and this is what mediates the fusion between neighboring cells, forming gigantic cells called syncytia. Okay, so syncytia is a group of fused cells with multinuclei called, again, syncytia. So these viruses bind to different types of receptors on the host cell surface, and this is mediated again by the viral attachment protein. And inside, of course, there is a nucleocapsid. We will start by talking about influenza viruses. So there are 4 types, as we mentioned, for parainfluenza viruses, 1 to 4, and under 4 we have 2 subtypes, 4A and 4B. Now these viruses can grow in cell culture, so we can really study them in labs. They can be grown on viro cells. These are monkey kidney epithelial cells, and they can also grow on LLC MK2 cells. This is just for your information. You don't have to memorize this. Just know that we can culture these viruses. What about the burden and epidemiology? So human parainfluenza viruses are the second leading cause of acute lower respiratory tract illness leading to hospitalization. So they are an important cause of hospitalization, particularly in children. HPIV1 2, and 3 are responsible for nearly 30% of the lower respiratory tract infections in children under 5 years of age, and 50% of croup. Croup is the type of horse cough. This is due to the development of laryngotracheal bronchitis. Okay, so parainfluenza viruses are second to RSV and third to influenza in terms of leading causes of acute lower respiratory tract infections. Human parainfluenza 4, virus 4, is rather rare compared to the other 3 types. In terms of seasonality, these viruses can be isolated throughout the year, but usually they peak in the spring or autumn, depending on the subtype. So I will show you here an example of circulation of parainfluenza viruses in Lebanon. This is a study that we performed a couple of years ago. It was published recently. And here we looked at the type of respiratory viruses associated with febrile illness in children with cancer. So here we're looking at around 16 viral types, and you can see most of these viruses circulate in the autumn to winter, particularly peak with a peak in winter. Okay, but sometimes we also see them early or late in summer, or late in summer or early in autumn. So here in pink we have the PIV1. We can see it in August, October. Now this is a small sample of patients. PIV3 is more common, at least for the during this time period of the surveillance. This was only a 1-year study, and then PIV2 also was detected during the winter season. Okay, here you can see also the Here I'm showing you a study that we performed a couple of years ago in Lebanon, whereby we investigated the incidence of different respiratory viruses, including parainfluenza viruses, in pediatric cancer patients. So we collected specimens from febrile patients throughout the year. This was a 1-year study, and we looked at about 16 viruses. So you can see here in pink, yellow, and gray the HPIV viruses. See in this study, PIV3 was most dominant, co-circulated with PIV1, and you see that in pink here the PIV1 was detected in August and October. Now, it's important to notice here that the most activity of these viruses, all respiratory viruses, is observed usually late autumn through early spring, right? But you can sometimes see some cases in summer, right? So you can use this as reference also for other respiratory viruses. We have coronaviruses, RSV, adenovirus, influenza B, and so on. And here I'm showing you just the incidence of mono-infection versus co-infection. This is important to know. Very often these viruses are associated with each other in infections. So in this study, the rate of co-infection was about 55%. Okay. So the human parainfluenza virus infects the epithelial cells lining the respiratory tract. Okay, so they are respiratory pathogens. So this is the natural route of infection. Thus, they transmit by respiratory droplets, and they cause rhinitis, pharyngitis, laryngitis, bronchiolitis, and pneumonia. And remember, croup is also distinctive with these viruses. Incubation period, we mentioned this before. Respiratory viruses usually have a rather short incubation period of time, particularly PIV, influenza, and RSV. In terms of symptoms, we mentioned these, but just to kind of reiterate, these viruses are associated again with rhinitis, pharyngitis, cough, croup, hoarseness, and fever that lasts for about 2 to 3 days. So it's rather a shorter duration of illness. We don't have any specific treatments as in antivirals or vaccines for these viruses, and thus the only treatment available is the symptomatic treatment. We can diagnose these viruses in the lab. We mentioned that we can culture them. This is not usually used for lab diagnosis. It's used could be used as confirmatory test or just to ensure that the virus we're detecting is infectious. Uh, More often now we're using PCR, but also immunofluorescent assays could be used. Respiratory syncytial virus is very frequent, particularly during winter weather around the world. It's actually the leading cause of acute lower respiratory tract infections in infants. Uh, it's been estimated that by the age of 2, nearly 100% of children will be at least infected once by RSV. Here's an example about RSV in Lebanon. So here first I want you to see the frequency of RSV-A and B. Now sometimes we cannot subtype the virus either for low titer or because of non-specificity of the primers. But as you can see, we start seeing RSV usually late autumn, and then it peaks in winter, and then by spring it starts going down. This is a very typical RSV season. So I had actually one of my volunteer medical students. He did this work and published it in a very reputable journal. So what he did, we had a lot of specimens in the lab, and he sequenced 45 samples from the season 2014-15 and 8 samples from 2015-16. And he also screened around 519 respiratory specimens from patients with influenza-like illness and found that 83 of them were positive for RSV. And then he sequenced these both the G genes and the F genes from these samples, and he genotyped them using genetic analysis phylogenetic trees. And he identified that actually in Lebanon multiple genotypes co-circulate each year. He identified this recently reported novel genotype ON1. He reported it for the first time in Lebanon. And you can see here there was an increasing frequency of this genotype in Lebanon. He also did the same for influenza B viruses. But what's interesting is that he was able to identify 2 new genotypes, which we named LBA1 and LBA2. These were, as you can see, they were very unique to Lebanon with a very strong support in this genetic tree. Okay, now just for those who don't know, who are not familiar with genetic trees, this is a tree comparing the genetic diversity between different viruses. So the longer the branch, the more genetic diversity you see between these viruses. You can refer to the paper for more details. Now, RSV infects the apical ciliated epithelial cells of the airways, okay, and thus interferes also with the ability of our respiratory system to clear mucus. As I'll show you later in our experiments in the lab, the infection is characterized by excessive mucus secretions, which contribute to difficulty or pulmonary obstruction. Okay, now the earlier that they happen in life, these infections could predispose the child for becoming asthmatic in the future. Here we're seeing HEP2 infected cells and uninfected cells, sorry, and here you're seeing the infected cells, and you can see these gigantic cells with multinuclei in them that we call syncytia. This is a respiratory virus, so needless to say, it's transmitted by respiratory droplets, and it has a relatively short incubation period, 4 to 5 days. Something I want you to know about incubation periods: these ranges can differ between textbook and another, and between one reference and another. Why? Because they're based on studies in different contexts, in different sometimes populations, but more or less they would just give you an idea about how short or long is the incubation period. And usually the average is used to indicate the incubation period. This is an experiment that one of my graduate students did in the lab. She developed a 3D culture model, or in vitro spheroids, of A549 cells. These are alveolar epithelial cells. And she infected them with our respiratory syncytial virus isolate 2001-2020. Here you see the DAPI, which is a nuclear strain. You see the stain for the virus, RSV F protein stain. This is done with labeled with antibodies, fluorescent antibodies. You can see the stain for mucin. The uninfected cells produce mucin, but no, you can see here there's significant increase in mucin production as cells are infected with RSV. Here you can see a successful infection of the spheroids as evident by the RSVF stain. You see that as we progress in time, the virus starts to penetrate into the spheroids. And here just a quantification of multiple experiments using the same setup. You can see a significant increase in mucin secretion. Again, this is a hallmark of RSV infection. So RSV symptoms are just like the symptoms of common cold, and they are really difficult to differentiate from other respiratory viral symptoms. Uh, we have runny nose, decreased appetite, particularly in children, cough, sneezing, and usually fever is a little bit lower in grade than, for example, influenza. And often in children, we see wheezing, which is due to bronchiolitis, and occasionally they can cause pneumonia. As I said, that RSV infection predisposes children to an asthma in the future, particularly if it happens very early on in life. Uh, the key signs in children, they don't have a lot of symptoms, but decreased activity and apnea might be the only ones that you will see. In adults, infections might go unnoticed or might be very mild, but we know now that RSV could cause also more severe disease in elderly. Some of the common symptoms in adults are runny nose, sore throat, cough, headache, fatigue, and fever. This is again from the study that I showed you earlier by one of my PhD students who is now graduated. So here we looked at the association between some of the common respiratory viruses and different variables. You can read this in details in the paper, but I wanted to show you here that RSV was particularly associated with co-infections. And in terms of outcome, you see that RSV was associated with sore throat as well as in some cases with diarrhea. Okay. Now the hospital admission or duration of hospital admission was relatively longer with RSV as well. Of interest, as I showed you earlier, RSV was also associated with bronchitis and bronchiolitis. Okay. So the darker the color towards green, the higher the association. For many years, when I taught this lecture to my students, I would tell them that, sadly, despite the high burden of RSV in children and the elderly, we do not have antivirals or vaccines that are effective to prevent and treat this illness. We still don't have direct-acting antivirals against RSV. However, thanks to the recent advancements in the understanding of the immunity of RSV infections, as well as the structure of the F protein, scientists have developed several effective vaccines for RSV. These vaccines have now been approved for adults greater than 60 years of age and also as maternal vaccination in pregnant women to protect infants from birth to 6 months of age. And these target the F protein. Another way to prevent the infection, particularly in the very young children who are at high risk of complications from RSV, is to actually do passive immunization or administering monoclonal neutralizing antibodies like the long-acting antibody that's now available and called nirsevimab. And this is administered ahead of the RSV season and provides protection for the whole duration of the RSV season. Now, we can diagnose RSV in the lab. RSV can actually be cultured. However, we don't use this as a way of diagnosis and management. But mostly we use real-time PCR as well as
rapid diagnostic kits such as the lateral flow devices. And also we can use immunofluorescent assays for detection of RSV.
Next, we will talk about measles or rubella. So this is different from rubella, and this is known as 9-day measles or al-hasba bil-Arabi. So 9-day measles opposed to the 3-day or German measles caused by rubella virus. So here's a question just to test your knowledge. Has measles been eradicated? Think about this and I will answer you in a bit. So unfortunately, measles has not been eradicated yet, despite the availability of a highly effective vaccine, as I'll tell you later. And in fact, recently we've been seeing resurgence of measles. This is partly because of the anti-vaccine movement, the incomplete vaccinations in some countries. This has resulted in failing our goal to eradicate measles. So let's see. Which of the following best describes measles? Think about it for some time. So measles, yes, in general, it can be a mild disease, but it do actually result in severe and sometimes fatal illness. So it's not always mild. It is preventable because we have a vaccine, and it can affect all children.
Measles continues to spread in countries around the world to various degrees. In fact, some countries have achieved measles eradication targets, or in other words, they managed to eradicate measles for many years. Although unfortunately, due to the vaccine hesitancy and anti-vaccine campaigns, we have been seeing drops in vaccination rates in some countries, leading to re-emergence of measles. And measles outbreaks are usually characterized by a 2 to 3 year cycle, meaning that we have outbreaks every 2 to 3 years, especially when vaccination coverage is not optimal. It's a very highly contagious disease. The R0, or the reproduction number of measles, is around 16 to 18, meaning every infected individual can infect up to 18 people in the society. If you compare this to a respiratory virus like influenza or COVID, where the reproduction number is somewhere between 1 to 3, then you could tell measles is actually a very contagious and transmissible virus. It can spread easily by contact with infected foam or with contaminated fomites, surfaces contaminated with respiratory secretions, or breathing air that has respiratory secretions from individuals who are infected. It has an incubation period of 1 to 2 weeks. Now, the good thing is that humans are the only reservoir or natural host of measles, meaning that if we can stop the spread of measles within humans, we can effectively eradicate the virus. There is no other source of the virus outside of humans. And the other good news is that we have one antigenic type of measles, again, which is very helpful when you think about vaccine effectiveness and our ability to eradicate the virus.
For some time, we had really gotten measles under a good control. However, we've been seeing a kind of resurge in measles outbreaks worldwide. So here you're looking at a heat map worldwide. The darker the color, the higher the number of measles cases per million. Okay, so we've seen major problems in Ukraine. Here you can see that they had almost 65,000 cases in Philippines, Madagascar, Yemen, Brazil, etc. In Lebanon, also, we've seen some resurgence of measles. In 2018, we recorded close to 1,000 cases in Lebanon compared to much less in the previous years. Here you're seeing also the trends of measles circulation between 2015 and 2019. So you're seeing in some regions, these are the WHO regions. This is here in orange, the Eastern Mediterranean region in which we belong. And you can see that some regions have been recording a striking increase in measles cases, particularly the Euro-region, the Afro region as well. We also had some increase in measles activity in our region as well. So measles infection progresses as follows. So after inhalation of the droplets containing the measles virus, the virus infects and multiplies in the respiratory tract. So here we're seeing here the timeline of progression of pathogenesis of measles. So the incubation period can be up to 2 weeks, as we mentioned earlier. The virus spreads from the respiratory tract to the regional lymph nodes. It replicates there, and then it causes a primary viremia, whereby it seeds the virus into the reticular endothelial system. The virus replicates there, and more or larger amounts of virus are produced, resulting in a secondary viremia that seeds the virus into other tissues like skin and conjunctiva. Okay, and this is where focal replication occurs of the virus. So here we're seeing again the timeline of events. In the period just few days preceding the rash, we have the prodromal symptoms, the respiratory symptoms, and the Koplik spots that are red spots that appear in the mouth. Around day 14, you have the appearance of rash, and this is associated with the production of or recruitment of CD8 T cells. So this is caused by this response. You also start seeing antibody IgM during the rash period. Now, the IgG antibodies kick in by the end of the rash period. Okay. Here we're looking at the disease where you have the prodromal symptoms and the rash, but the infectious virus is present in the blood before the appearance of symptoms and during the rash period. Now, 5 to 15 years after the initial infection with measles, some patients develop subacute sclerosing panencephalitis, which is a lethal disease that we'll discuss in a little bit. It's important to note that measles can also replicate in certain lymphocytes, so it can replicate in these cells that helps in the dissemination of the virus throughout the body. So again, before the rash appearance, we have the prodromal symptoms, which are usually respiratory symptoms associated with very high fever that lasts 4 to 7 days. You have runny nose, cough, red and watery eyes, and then you have these Koplik spots, the red spots with a white center that appears inside the cheeks, inside the mouth. Okay, this is followed by a rash that usually appears on the face and upper neck. Okay, so the rash usually appears on the face and upper neck, and in about 3 days the rash starts spreading, reaching the hands and feet as well. Right, so this is here the typical measles rash. So complications of measles include otitis media and pneumonia. It's important to note here that measles infection is associated with severe immune suppression in patients, and this facilitates secondary bacterial infections. And for instance, in patients with latent tuberculosis, this could result in the exacerbation of disease. Blindness and as well as encephalitis can occur, but these are rare events. As we mentioned earlier, 5 to 15 years after the initial infection with measles, some patients develop subacute sclerosing panencephalitis. This is a rare complication that happens in 1 to 10,000 or 100,000 cases. However, it's a very debilitating disease and could end up in death after 1 to 3 years. SSPE is characterized by progressive mental illness, deterioration, involuntary movements, as well as muscular rigidity and coma. So measles can be easily diagnosed based on clinical evaluation and serological testing. Remember, we can see IgM during the rash period, and also, of course, by real-time PCR or virus culture. Now, the clinical value of diagnosis is important just to prevent spread of the disease, but we don't have a treatment that's specific for measles. The vaccine is available. It's again similar to the 3 days measles vaccine, the MMR vaccine, and in some cases it can be combined with varicella. The vaccine is highly effective and provides long-term immunity, and as we mentioned before, it's administered at the first dose around 1 year of age, 1 to 1.5 years of age, and then the second dose at 4 to 6 years of age. Okay. So in 2012, the WHO put a strategic plan to end measles worldwide, to eradicate measles. By 2015, their goal was to reduce the global measles death by around 95% compared to the 2000 levels, and to achieve at least regional. It means that, for example, if we take our region, the Eastern Mediterranean region, they wanted to achieve regional measles and rubella elimination goals in some regions. The goal was that by the end of 2020, we would have achieved measles and rubella elimination in at least 5 WHO regions.
Measles eradication strategies focus on 5 main pillars. The most important of these is achieving high vaccination coverage among the population, particularly, of course, among children, and full vaccination, meaning 2 doses of the vaccine. And this is because, as mentioned earlier, measles is a very highly contagious infection, and thus it's important to attain close to universal vaccination coverage to break the transmission cycle of the disease and reach that eradication target. And to support also this, we obviously need to monitor and understand the spread of the virus so that we can quickly detect any outbreaks, and this is through effective disease surveillance and reporting. And this allows us to achieve the third pillar, which is really responding to any outbreaks very well so that we can prevent them from spreading further. And this is what we call outbreak preparedness. So, for example, in case of an outbreak, we can think about education campaigns to educate people about how measles spread, and also to do any catch-up vaccinations, so to vaccinate kids who have missed their vaccines. And this is linked also to, as I mentioned earlier, we need to have very strong public awareness and education, particularly in communities where we're seeing hesitancy around the value of vaccinations or the importance of preventing measles. And thus we need good public awareness and education. And also we need to invest also in better tools to quickly detect and respond to any outbreaks.
So next we will talk about mumps, النكاف بالعربي أو أبو كعب بالعامية. Mumps is also known as epidemic parotitis. This is the key distinguishing symptom of mumps. Parotitis means the inflammation of the parotid gland. The distribution of this illness is global, and its incidence is greatest in winter and spring. The virus reservoir is human, and it has one antigenic type, and this is, as you remember, very important for controlling and eradicating the disease. The virus can be transmitted via respiratory droplets, and it has an incubation period of around 2 weeks. Now, patients infected with mumps can be contagious even before the appearance of the symptoms. So, they can be infectious in the prodromal period. This is similar also to measles. So as with the other respiratory viruses that we've seen so far, the virus replication starts in the respiratory tract. We have local replication, we have viremia, and systemic infection. The virus then disseminates to the parotid gland, and it multiplies in the ductal epithelial cells, causing local inflammation with marked swelling. And this is why we see this futuristic parotitis, or what we call in Arabic Abu Kab. The virus can also go to the testis, ovaries, peripheral nerves, eye, inner ear, central nervous system, causing also inflammation there. It can also uh, infect the pancreas, and this could be associated with onset of juvenile diabetes. So what are the clinical manifestations of mumps? Often the infection can be asymptomatic or mildly symptomatic. In symptomatic people or children, we can see high onset or sudden onset of fever, malaise, and anorexia. The illness can manifest as parotitis, which is a uni or bilateral inflammation of the parotid gland accompanied by fever in almost 50% of the cases. This is the parotid gland, and you can see the 6-year-old girl has unilateral parotitis. Inflammation of salivary glands and pancreatitis can also occur. Orchitis can occur, which is inflammation of the testis. So also orphritis can occur, which is inflammation of ovaries, aseptic meningitis, and encephalitis. In addition to mumps, Coxsackie virus and echovirus are among the top 3 viruses associated with aseptic meningitis. And aseptic meningitis means that this is not a bacterial caused meningitis. Diagnosis mainly relies on clinical presentation and evaluation of the patient, particularly the parotitis. Also, we can detect the virus by PCR. Serological testing can be particularly useful to detect exposure or previous infection. We don't have specific treatments for mumps like with the other viruses that we've described, but we can for sure do symptomatic treatments, so treatment of symptoms, fever, and inflammation. For prevention, we have the famous MMR vaccine, the measles, mumps, rubella vaccine. That is a live attenuated vaccine, and it's very effective in preventing this disease.